Good evening. First, I would like to thank Dean Toller and Dean Crawford for this great honor, and thank you all for being here this evening. I will give you a quick tour of my research program on structural biology of virus entry and antiviral development, what we have learned, and where we are headed. In my PhD study at Yale, I worked with Nobel laureate Dr. Tom Stites. There, I determined the first crystal structure of the tRNA-CCA adding enzyme. This enzyme is unusual. It adds the CCA sequence to the 3' end of tRNA molecules without using a nucleic acid template. My study showed that the protein itself serves as a template for adding the CCA sequence. After my PhD, I wanted to work on translational research. I was deciding between cancer and virology. Then SARS-CoV-1 hit. It caused more than 8,000 infections with a 10% fatality rate. That made my choice clear. So I joined Harvard Medical School as a postdoc and worked with Dr. Steve Harrison, who is a pioneer in structural virology. Because the spike protein on virus surface drives viral entry into host cells, I began to study the structure and function of the SARS-CoV-1 spike protein. This launched my career in coronavirus entry. The spike protein is a homotrimer on the virus surface. It contains 3 copies each of a receptor binding S1 subunit and a membrane fusion S2 subunit. During viral entry, S1 binds to a host cell surface receptor for viral attachment. The spike protein is cleaved by a host protease at the S1-S2 boundary. Then S1 dissociates and S2 undergoes a dramatic structural change to fuse the viral and host membranes. The spike protein is present in 2 states, pre-fusion and post-fusion. It plays key roles in viral infection, pathogenesis, host range, and tissue tropism. It also induces most of the host immune responses and is a primary target for vaccines and antiviral therapeutics. To block viral entry, we can block receptor binding, protease cleavage, or membrane fusion. I collected the first EM images of the SARS-CoV-1 spike protein in the pre-fusion and post-fusion states. At that time, EM was not as advanced as nowadays, so these images were at low resolutions. I also provided the first direct visualization of the spike protein's conformational change. I further collected EM images of the SARS-CoV-1 spike protein complex with its ACE2 receptor. ACE2 binds to the tip of the spike trimer. That is where the receptor binding domain, or RBD, is located. I then determined the crystal structure of SARS-CoV-1 RBD complexed with human ACE2. This was the first atomic structure of a coronavirus spike protein. It opened a new era in structural biology of coronavirus entry. The RBD contains 2 subdomains, a core structure and a receptor binding motif, or RBM. The RBM mediates viral interactions with the ACE2 receptor. In 2007, I moved to Minnesota as assistant professor. My lab continued to work on the RBD ACE2 interactions. We determined the molecular mechanisms for the cross-species transmission of SARS-CoV-1. Specifically, we showed how 2 RBM mutations allow the virus to transmit from its animal host, Palm civis, to humans. In the meanwhile, we started to work on the entry mechanism of many other coronaviruses. Coronavirus spike proteins are very diverse, and they recognize a wide variety of host receptors. Our research showed how distinct RBDs can recognize the same receptor, how similar RBDs can recognize different receptors, and how coronaviruses evolved to achieve this. We also showed that coronaviruses could hijack host proteins and use them as viral RBDs. Our research was the first to show that host protease activities can function as a barrier for the cross-species transmission of coronaviruses, in addition to RBD receptor interactions, which functions as another barrier. Furthermore, we did structure-guided RBD vaccine design and developed a highly effective RBD vaccine against the MERS coronavirus. In 2017, we determined Minnesota's first cryo-EM structure. It was a pig coronavirus spike protein. In late 2019, SARS-CoV-2 hit. In January 2020, the genomic sequence of SARS-CoV-2 was released. On the same night, I stayed up and drafted a manuscript to make early predictions of the SARS-CoV-2's molecular behavior. These predictions include AC2 is a receptor for SARS-CoV-2. What animals can be infected by SARS-CoV-2 and what animals cannot be infected? We also predicted the AC2 mutations in mice and rats that allow SARS-CoV-2 to infect these animals. This lays the foundation to make small animal models for SARS-CoV-2. We also predicted evolution of key RBD residues. All of these predictions were later confirmed experimentally. Also in early 2020, we identified 3 unique structure features of the SARS-CoV-2 spike protein. First, SARS-CoV-2 RBD binds human ACE2 more strongly than SARS-CoV-1 RBD does. Second, in the trimeric spike protein, the RBD is present in 2 different conformations, standing up for receptor binding and lying down for immune evasion. For SARS-CoV-2, the RBD is hidden about 50% of the time. This allows the virus to balance infectivity and immune evasion. Third, a human protease called fusion cleaves SARS-CoV-2 spike protein. These 3 mechanisms allow SARS-CoV-2 to spread widely, causing COVID-19. All of these findings are now widely accepted in virology. During the pandemic, we continued to work on the structure and function of SARS-CoV-2 spike protein. We identified a molecular switch in the spike protein that regulates the up and down conformations of the RBD. We identified unique structure features of the Omicron spike protein, which allowed Omicron to spread even more widely than the original SARS-CoV-2. We pioneered an approach called structure-guided virus hunting to understand cross-species transmission and potential evolutionary origins of SARS-CoV-2 and Omicron. Our approach is now being widely used in virology. Overall, our research determined the most fundamental molecular mechanisms for the COVID-19 pandemic and explained where SARS-CoV-2 and Omicron came from and why they spread so widely. In the past 2 decades or so, my lab has determined many structures of coronavirus spike proteins. In fact, we determined the first 7 structures reported in the field. By determining these structures, we established the field of structure biology of coronavirus entry and defined key structure biology principles that have guided global COVID-19 research. In 2022, we received NIH funding to establish the Midwest Antiviral Drug Discovery Center. This is the largest NIH grant in UMN history. Our center involves 18 institutions and more than 40 academic labs. I serve as a co-director of the center. We develop antivirals against viruses with pandemic potential, including SARS-CoV-2, Ebola, Marburg, dengue, and Lassa. Our goal is to develop next-generation antivirals that do not require injection or refrigeration. Importantly, these antivirals need to be adaptable to viral mutations, which is an important lesson we learned from COVID-19. We work on 2 types of antivirals. One is the inhalable nanobodies, which are single-domain antibodies from camelid animals. The other type is orally available small molecule compounds. So far, we have developed 9 nanobodies that target the SARS-CoV-2 spike protein. They are named Minnesota series in honor of the University of Minnesota. Most of these nanobodies bind to the RBD and directly block AC2 binding. Among them, Minnesota 9 is the most up-to-date against current Omicron subvariants. A few of these nanobodies bind to regions outside the RBD. Minnesota 5 is interesting because it induces RBD to close, which indirectly blocks AC2 binding. It also blocks fusion cleavage. Importantly, it targets a conserved epitope that is not accessible to human antibodies. Overall, nanobodies have many advantages over human antibodies as antiviral therapeutics. They are much smaller, which gives them superior epitope accessibility and tissue permeability. They also have high stability and do not require refrigeration, and they are compatible with inhalation delivery. While we were developing these nanobodies, we also developed a novel approach called structure-guided nanobody evolution to overcome viral escape mutations. The idea is that if viruses evolve, nanobodies can also evolve to keep pace with the viruses. To do that, we first determine the structure of the viral target complex with the nanobody. We then identify the viral mutations at the interface and select nanobody residues that surround these viral mutations. We then completely randomize these selected nanobody residues and build a phage library that displays all of these mutant nanobodies. We then select those mutant nanobodies that bind to the mutant viral target with high affinity. The whole process can be completed in less than 2 weeks. It is much faster and just as effective as natural immune responses in tackling new viral variants. Importantly, our approach enhances existing nanobody therapeutics without discarding them. This is in contrast to human antibodies, which are discarded once viral escape mutations occur. Our goal is to update, do not rediscover antiviral therapeutics. Our approach can save huge amount of research resources and also greatly shorten response time to new viral variants. We have expanded our research to other pandemic viruses. Ebola and Marburg viruses are highly lethal, and they cause recurrent outbreaks from animal reservoirs. Both dengue and Lassa viruses cause millions of infections and thousands of deaths each year. All of these viruses pose serious threat to global health. Against these viruses, countermeasures are either non-existent or have serious limitations. We use the same structural biology platforms to study these viruses, from atomic understanding of viral entry to developing novel nanobodies and small molecule compounds to block viral entry. So far, we have developed inhibitors against all of these viruses. Our entry inhibitors against Ebola and Marburg are particularly advanced. We developed the first 2 nanobodies targeting Ebola. We named them Nanosota EB1 and EB2. We also developed the first 10 antibodies against Marburg. In the meanwhile, we have developed a small molecule inhibitor that targets Ebola. This is the first orally active Ebola small molecule inhibitor. We are very excited about these inhibitors and hope to push them to clinical trials. To sum up, we established the field of structural biology of coronavirus entry and pioneered the field of structure-guided virus hunting. Our research identified key structural mechanisms of SARS-CoV-2, which have guided global pandemic research. We are now studying the entry mechanisms of other pandemic viruses and tracking their cross-species transmission. We are eager to translate our atomic understanding of viral entry to novel antiviral therapeutics. Our goal is to build a versatile, rapid response antiviral platform for pandemics and biodefense, so the next pandemic can be prevented, or at least its impact could be dramatically reduced. This award I am receiving reflects teamwork. I would like to thank all of my graduate students, postdocs, technicians, and undergraduates for their hard and creative work. I want to thank our collaborators for validating our structural biology findings using viruses and for testing our antivirals in animal models. I'm grateful to everyone who has worked with me throughout my career. Thanks to NIH for funding, and thank you for your attention.